Dihybrid crosses work in all the same ways and can be used with any of the crosses that we've already discussed. The only difference with them is that we are going to be focusing on two traits at the same time. So what we have to make sure of is that we take special care when we determine the gametes, determine what each parent could possibly be passing down. So let's take a look here. We're going to be talking about pea plants, and we're going to talk about their coloring as well as their height. Okay, those are our two traits. So the first thing we need to do is go through and actually create the key, just like normal. We are told that green is dominant to yellow. So we'll use a big G to mark green and a little g for yellow. And that tall is dominant to short, big T, little t. So let's take a look at this first practice problem. We need to cross a heterozygous green, heterozygous tall plant. So its genotype is going to be big G, big T, little t. It's heterozygous for green and heterozygous for tall. We're going to be crossing that with another heterozygous green, heterozygous tall plant. This next portion is called determining the gametes. In the past, we've always just been able to separate these two and say, okay, the parent would pass down either a big G or a small g. But now, since we are going to be taking a look at the differences in these two traits, we have to be a little bit more concerned. We're going to use a method called FOIL. It stands for first, outside, inside, and last, just like in math class. So in order to determine the gametes, we're first going to take the first two genes. So we would see one gamete's going to be big G, big T. This parent plant could pass down a big G and a big T. Then we're going to take a look at the outsides. Big G, little t. The insides, little g, big T. And the lasts, little g, little t. In doing this, we have found the 4 different types of possible combinations that this parent could pass down. Let's do the same thing again over here on this parent. Remember, we're going to take the firsts, big G, big T, the outsides, big G, little t, the insides, little g, big T, and lasts, little g, little t. Now these gametes are going to be what get filled in along the top and the sides of the Punnett square. Since each parent can pass down 4 different types of gametes, we have to have a Punnett square that's 4 by 4. So let me make that really quick. Okay, so we're going to plug in here big G, big T. Then I'm going to plug in my big G, little t, little g, big T, and little g, little t. Then I'm going to plug in these gametes along the top. Big G, big T, big G, little t, little g, big T, and little g, little t. I'm going to start filling in the box. Now, as I do, we're going to run out of time on this video, and when we pick up in the next video, you'll see it's totally completed. All I'm doing is I'm saying big G, big G, big T, big T, big G, big T, little t, and so forth. Filling in the Punnett square, just like we've always done, seeing what combines together. See you in a minute.